I accepted this invitation with some reluctance. It's sort of, excuse the expression, end of the road kind of talk, and I'm not really intending to quit cold turkey myself. But on the other hand, I thought I might not get another chance. At this age, it's sort of like playing blackjack, and who knows what will happen tomorrow. So I grabbed it. And I'm going to tell you about some of the things that John mentioned, and especially life before bioinformatics. Bioinformatics is big now, but it didn't exist when I came to UCSD and joined John back in 1964. It's something recent, and we can show this by a little graph here, citations in PubMed to the term bioinformatics. Now, the first citation appeared in 1990, not surprisingly by the people who invented the word back at the NCBI. And you can see that it took a while to catch on, but then all of a sudden the rush came. Every department had to have a bioinformaticist. Every school had to have a bioinformatics department, industry. The question is this enduring? You'll notice that the graph here only goes to 2002. If we put last year in, you can now see that maybe it's a passing fad. On the other hand, maybe it's just that people are getting used to the term and they don't feel they don't have to use it so much anymore. So if you're a bioinformatics major out there, don't change yet. But on the other hand, keep those laboratory skills honed up. You never can tell. I'm going to tell you about one of the route paths that led to the field of bioinformatics. My talk will not go past the introduction of the term. It's going to end in the late 1980s. And there are many different ways that this field got started. And I'm going to commit the sin of all historians of neglecting the many to mention a few. And so I'm going to tell you some of the people that contributed greatly to the swell that was to develop that became, I think, the notion of gene searching especially is what we're going to be after here. Now, two of these people that led this charge were in fact chemists. Fred Sanger, who was destined to win 2 Nobel Prizes, had a reagent named after him, the Sanger reagent. There was actually fluorodinitrobenzene by name. And Edmund also had a reagent named after him, phenylisothiocyanate. Both of these guys actually went to the literature and took already existing compounds and changed one atom and made them much more efficient and functionally useful. But the point is, these guys started and gave the ability for people to go out and get digitized information in the form of amino acid sequences. You got them one step at a time with the Edmund degradation, and it was marvelous. So in the early 1960s, there came to be what seemed like a plethora of data. Hundreds of amino acid sequences. There are going to be two of the people. I'm going to now mention some of the others. If they had done it in a vacuum, it wouldn't have gotten any place. But there were also zealots. These guys were very early molecular evolutionists, and I put the ones on the slide here that I'm likely to mention later. Morris Goodman, and Morris is one of the people who actually started the interest in what you might call anthropoid molecular evolution. Tom Jukes, a wild man, but a very big spur in the field of molecular evolution. Alan Wilson I'm going to mention a little bit more, especially when I get to chimps myself. And Emil Zuckerkandl, who had begun as a postdoc at Caltech, working with Linus Pauling, but had to spend his entire career working in the area of molecular evolution and provided the wherewithal, a new journal, for example, he started, Journal of Molecular Evolution, that allowed all these data to pour in and be thought about, all getting ready to become later bioinformatic. Besides these guys, there had to be a couple of amateur computer guys. I'm sure that the people in the computer field will, of course, be thinking back of information theory and the like, but a couple of people who got this started in terms of the biology were Margaret Dayhoff and Walter Fitch. Margaret, who died prematurely back in the 1980s, had started, and jointly with a couple of others, what was called the Atlas of Protein Sequence and Structure. And it was the gen bank of the 1960s and 70s, you might say, but of course for proteins and not for nucleic acid. A very, very important development that let everybody see what kind of data were available. Walter Fitch had some really important papers in the Journal of Molecular Biology in the 60s about lining up sequences and what it might mean. And he did this with some very simple and enduring computer algorithms. Then there were a couple of guys who actually wrote what we call influential monographs. Susumu Ono, who wrote a book in 1967 extolling the virtues of gene duplication. Now, much of the talk today is about gene duplication. Gene duplication was well known before this book, but this gave everybody an easy source to cite in the ensuing 30 or 40 years. But it put it all together, and it convinced a lot of people, hey, this thing can really get going here. Kimura wrote a book in 1968 on the neutral theory of evolution. And these two things together now are going to have to do with gene duplication and then amino acid replacement, and the fact that many of these amino acid replacements are neutral in terms of not having and yet become the trail markers of the things we're after here. This was really quite an interesting thing. There were classical geneticists on this campus who refused to accept the notion of the neutral mutation. What Kimura had pointed out was that at any given moment, of all the copies of a gene that are in existence in a given population, only one is destined to be around to spawn all the copies of that gene in the future. And the mutant one, if it doesn't have any plus or minus effect, is just as likely to be that one as any other. And yet there were people who were so schooled in the notion of survival of the fittest, they just couldn't believe that this was the way it was going to work. So I mention them because Darwinian evolution in terms of gene duplication followed by amino acid replacement or base modification, that's what's the theme of what we're going to be talking about here. Well, will we ever get through it, though? All right, well, this is a really important table that appeared in a book in 1962. And it's from Emil Zuckerkandl and Linus Pauling. And it was a chapter in a book about hemoglobins. And they pointed out that there were, by this time, several hemoglobin sequences known, mostly as a result of work of Vernon Ingram for one, and Braunitzer in Germany for another. But these guys looked at it and counted up the number of differences between the human hemoglobin, for example, human hemoglobins, and gorilla. And they also did the same for the alpha versus beta chains. And they made some estimates about how long ago these sequences must have diverged. Now, this is the first citing I know of where molecular data were used to look into the past in this very formal way. I know about it very much because, you see, until I knew about this paper, I thought that I, in fact, had put the first business in the record of sequence divergences correlated with geological time. This was a paper with Bloombeck in 1964, and it's not hemoglobins, it's fibrinopeptides. And we had looked at a number of species, and by comparing it with when the paleontologist told us that these diverged, we were able to make this graph. Later, somebody pointed out this earlier one, and it certainly was right there, but I was oblivious at the time. But obviously, different molecules, it's okay. Everybody can't be first. Fibrinopeptides, you can just, these are all ancient slides. You can tell that the single letter code hadn't yet emerged. People typed out these three-letter cognates. But I was, as mentioned, a postdoc in Sweden. And so we had a chance to do the usual domestic creatures of cow, sheep, and pig. But we also were able to do reindeer, and which seemed a curiosity at the time. But you must always take advantage of your opportunities. And it turns out that all of those, except the human up there, the top four, are all cloven-hooved animals. They are artiodactyls. And with a head start on this, I thought, why don't we do just a ton of artiodactyls and see if we can account for every possible amino acid replacement as the the radiation occurred. I wrote actually to George Gaylord Simpson, who was the leading paleontologist of the day, exchanged several letters with him, told him what we were doing, and he gave me his best estimates about when, for example, the deer diverged from the giraffes, et cetera, et cetera. He told me, quite frankly, he didn't believe in these molecular data. I had explained that these fibrinopeptides, because they were just out there on the surface of the molecule as sort of an electric fence, could be almost any amino acid. There were a few that couldn't. And so they turned over very, very rapidly. And he thought, oh, that sounded terrible. It must be awfully random. But in fact, it was a very good one for charting the course here. And here is a study of those creatures now. This was done here in Bonner Hall, most of these guys. George Moros was a graduate student in biology back in the 60s. We had the San Diego Zoo. We would leave our bottles down there with anticoagulant, and every time they'd bring in some buffalo to have its toenails clipped or something, we were able to get some blood, and we assiduously purified these peptides and sequenced them. And although we couldn't get every single replacement, we could get most. In other words, every place you see an A and a number, it tells you a particular place in the sequence where an amino acid replacement had occurred on the way to this radiation. And it was pretty good. However, we also realized that the way to do this was to let a computer make these judgments. And I had another graduate student at the time, Susan Tiedemann, who was trained in biophysics and quite good at the computing, which was a very primitive field at the time. At any rate, Susan took it on to try and figure out how we could reconstruct this tree with the computer. Until now, what we're doing here is we're taking the paleontologist's word for it and just plugging in these values with the tree that they told us was true, which for the most part was very accurate, of course. Susan got it to the point where she could make a matrix. And we had some, I don't know, there may be 19 different species or something like that in this tree. And we could now count all the base substitutions that it would take to account for those amino acids in all the different guys. But we were stumped. We didn't know what to do next. And it was Walter Fitch, who I mentioned earlier, who in fact broke the logjam in 1967, one of the first biological algorithms. Fitch showed how to make a phylogenetic tree with sequence data. It turned out to be really quite simple. He made a matrix like the one I had just shown, and then you take the two taxa in there that are the closest and you just combine them and average them and then make another matrix, and so on and so on and so on until you fill up the whole tree. This happened to be for the protein cytochrome C, and it was a marvelous thing. Cytochrome C is a slow changer, and as a result, he didn't confine himself to just one group of mammals the way we were. He could take this all the way down to Neurospora and Candida. It was a marvelous achievement. 1967, first real good computer-generated tree. Margaret Dayhoff had some competition going at the same time, but this one was so simple and made available to everybody. It was good. Fitch actually came here to work in my lab in the late 60s so that he could feel as though he could get his hands wet also. After the month, I think he went back to the computer and stayed there. It's much harder at that time. Getting one residue a day was a very slow approach. Well, after we finished the artiodactyls, the big rage was to look at primates. I mentioned Morris Goodman. I had met him when I was a postdoc, and Alan Wilson. Alan had made a big splurge in the literature. He and Vincent Sarich at Berkeley had shown that using immunologic data, actually antibodies to albumin, and shown that by their reckoning, when they quantified this, that chimps and humans had diverged much more recently than people had thought at the time. At the time, people were thinking that chimps and humans maybe diverged 15 million years ago. You notice, you probably couldn't see, but on the Zuckerkandl and Pauling table, they had gorillas and humans at around 17 million years. Well, Alan announced brazenly that, oh, it's really only about 5 million years ago, and here are the data. Well, the first couple of sequences that were done or cytochrome C for humans and for chimps, and they were identical, but cytochrome C is a very slow-changing protein. That's why Walter Fitch was able to look back so far. Hemoglobins, it turns out that although there was one change for gorilla versus human, the hemoglobins were exactly the same. And so he said, well, why don't you do the fibrinopeptides? Because they change so rapidly. Certainly there must be some changes of the fibrinopeptides from chimp to human. So we obliged, and if you look up here on this, you can see that in fact chimp, gorilla, and man are identical. The two replacements that separate them from orangutan occurred before the trivergence there. So here, in fact, is an old tally of all the known sequences in someplace around 1968 for chimps versus human and some other species. And you can see that row of zeros going across the top. The fibrinopeptides, there were 40 residues involved there, all identical. Then there was the hemoglobin alpha chain, they were all identical. Hemoglobin beta chain, cytochrome C. These sum up to some 340 amino acids or thereabouts, I think, when we put them all together. Those are their lengths that are in parentheses. And they're all identical. A rumor began to get around that all the differences were going to turn out to be cultural. But in fact, a fifth sequence was done, carbonic anhydrase, and although it had 259 amino acids, there in fact was one difference between chimp and human. So very, very close. And people were beginning to feel either interested or threatened by all this data now that were coming out. All right, well, all of this has to do with a difference in sequences so far between species. But as I mentioned, the big deal was the fact that genes duplicate. They not only change, but you wouldn't get any place if you just all had one set of genes and they all just changed, but it's the fact that the genes duplicate and give you new proteins to work with. So here is just a simple little scheme of the day. It was realized that there are all these mutations that must be occurring somewhat randomly and fairly constantly, but only some small fraction of them was in fact surviving. And the fraction that survived would be different for different proteins. Some proteins are even different parts of proteins. Some would have high fractions because of the parts being very tolerant to insult. Others would just be absolutely refractory, and so there'd be slow chain. But anyway, you made your tree by counting up all the ones that actually had become fixed as opposed to just those that occurred. Well, you can make a companion tree for this, for duplications, that works exactly the same way, except the nodes, instead of being divergences where one founder guy went across a river and started a new species or something, would in fact be the gene duplication. But it's going to be the same thing where most of the mutations that occur are going to be lead you to some bad ending, but some will succeed and you're going to be able to develop new proteins. Now, there were a lot of gene duplications that were already known long before either the book or the computer. Some of these were small polypeptide hormones, for example, vasopressin and oxytocin. It was realized that they must have come from some common source. There were only 1 or 2 amino acid differences between them, even though they did different things. Hemoglobin chains were well known. In 1957, it was clear that they must have come from some common ancestor, alpha chains and beta chains and gamma chains. The clotting factors, I myself was very interested in blood clotting, and they were all coming from some common ancestral type, etc. I'm going to give you one example of this. It's the bottom one, immunoglobulin chains. And of course, I'm telling you about this one because it's going to give you some feeling for the reality. Some of these things become magnified in memory, and they sound so much better than they really were. And some of them were pretty dicey and risky. And so I'm going to show you this next one, but it was a good bet, as it turned out. Anyway, these are some partial sequences of the various chains of immunoglobulins back as they were in around 1965. Paper finally got published in 1966. So I'm just going to read a couple at the top. There's one sequence there that in a light chain of a gamma globulin, the sequence is pro-pro-ser. And then there's another sequence that came from a heavy chain, and the sequence is pro-pro-ser. Well, I thought about this, gee, that's really interesting. I took it in and I showed it to John. Now I have to tell you a story about John, which he alluded to earlier. John was originally trained as a physical chemist, and he took up biology late in life. I myself was a biology major, and so I had to learn all this other hard stuff later. However, physical chemists have a different time scale than the rest of us. They think in terms of milliseconds and microseconds, seconds occasionally. All right, so I showed the thing. I went, John, look at this. And he looked at, he said, sir. He said, why, that couldn't happen by chance in 1,000,000 years. I said, well, John, actually, we're thinking about 100 million years here, right? But different time scale. Millions of years to the evolutionists are just a tick of the talk here. I mean, this is nothing. Well, that was a lead balloon there. But anyway, here is what came out of it. On the basis of this and other data that we had at hand, we proposed that, in fact, antibodies were divided into domains. I mean, this is the first, I really think this is the first depiction of this ever. And everybody knew about light chains and heavy chains, people knew there were variable regions in both. But in fact, where we thought that these must have evolved by a series of gene duplications. And it turned out to be true. And we went on and did other experiments, especially some with Henry Metzger, to prove this. But once again, the thing to carry home is that people are changing their way of thinking about things, that genes duplicate, and they just begin to radiate. The old notion of radiation is a true one in molecular biology. So the point of this is, where do new proteins come from? New proteins come from the duplication of the genes for old ones. Over and over, again, had to be trying to sell this theme to people who mainly weren't just interested at the beginning, but then later were either accepting it very rapidly or not. It's going to be a point I'd like to make later. All right, where are we? We're swooping up here now. 1978, we're about to have that big change in life. Cloning is about to enter the scene. I mentioned that Margaret Dayhoff and her colleagues had an atlas of protein sequence. I'm going to tell you about that in a moment. But they had never foreseen that it was going to be Sanger, primarily, who would introduce DNA sequencing, and you're going to get all these tremendous protein sequences by way of their DNA. It was just unthinkable. I mean, it was thought that DNA just had these little 4 signposts. You'd never be able to figure anything out about it. It also became clear that if you're going to really get serious about this, you had to get into computers big. Now, by this time, I had been wading into computers more and more. And although I'll thank some people at the end, I must tell you that I had 2 very big helps getting into this in the early 60s, and they were my sons. It turns out that it wasn't that they were so filially loyal, but I had tended to be one of those overbearing fathers who said, why don't you help me with this sort of thing? And they were wide apart, and I pressed them both into service. They came along, one of them especially, when, for the short period of our public education system, when the new math was introduced. Now, the new math was really great. And I didn't understand it at all, but my sons explained it to me, and they got into this and really started helping. So I'll tell you some of the things they did later as we go here. All right, so here is now the atlas back in Washington, where you send all your sequence data after you worked hard to get all this, and then they would work on it and annotate it and keep it for what seemed to be endless periods, and put out periodic issuances, and very, very important service, no doubt. But not only that, if they found something interesting with your data, you were likely to read about it in Science someplace. So in 1979, it only had, you could buy a tape from them for $50, and it only had 1,081 sequences. But worst, it was a very redundant database. I realized this when we had just gotten a computer, little PDP-11, and we were trying to look at the frequency of di and tri and tetrapeptides, and some of these peptides were just overwhelming. There was one tripeptide, gly-cis-his, that occurred, it seems like a zillion times more than it should by random. And the reason it was that it was in every single one of those cytochrome C, which was in fact 10% of the database. I thought, well, we're never going to be able to learn anything about proteins. This is too prejudiced. We're going to start our own collection. And that was where my second son came in. He was, in fact, at the time 11, and I was in charge of him when he got out of school. So I had him learn to type in sequences from the literature. And he is an excellent typist to this day. I mean, he is very, very good. I also had my secretary, Karen Anderson, wonderfully, she took to this like water, and the two of them would put in these sequences, and they had to be all verified, and we made folders. We made our own atlas, and so we called this NUAT at the time. This was back in 1981 when we first had it. And we divided it biologically, and we got our sequences in two ways. First, we got it fresh from the literature. Had a lot of subscriptions to the standard journals. As soon as any sequence came in that was interesting, type it right into the database. Furthermore, we'll skip all the ones that are uninteresting. No immunoglobulins, no hemoglobins, enough already. Certainly no cytochrome Cs. Get them out of here. But anything else, in it went. Now you can see some prejudices of the day. You can excuse the anthropomorphism for humans, that would be one category. But many of the bacterial sequences were E. coli, and so it had its own bin. There were so many, what seemed to be a lot coming in at the time. All right, well, what we did, we had a regimen. The first is that we put them in, made a folder, and had some citation source where it came from. But we did one more very important thing. As soon as we got it, we searched it against every possible other sequence, our own database and the Dayhoff database. And as a result, it became literally a connection machine. Suddenly we were finding that there are all sorts of things that are related to all sorts of things that nobody ever thought were going to be related. So I'm going to give you a couple of examples. Well, the word got around. We did one other thing that was really quite noble in retrospect. We made the sequence searching service available for nothing to anybody who wanted. And in fact, during the early 1980s, we searched sequences, well, what seemed like for most of the country, but amounted to 400 or 500 individuals. I'm going to show you just a front page of a list in a moment. It's amazing the names that are on it. People would call up and say, will you search my new sequence? And we did it, as I say, nobly, in the sense that if they didn't want it made public, we were sort of like Swiss bankers. We would just keep it. There was one condition. If we searched it, they had to leave the sequence in the database. So that would be value added, you might say. All right, so everyone can. But if they said put a flag on it, don't tell anybody else, we made an agreement that we wouldn't tell them either unless both parties were willing to find out what had been the connection between. It really worked very well. Okay, so here is this list. Obviously, you can't read it, but I can look up there, and I'm just going to read you some names that are everyday household words in the LA Times. I see up here David Baltimore, Sam Barandas, Mike Bishop. I mean, just read on down here. Roy Britton. There are just everybody. Anybody who was sequencing at the time, they would send the things around. All right, I'm going to tell you now about some of the connections that were made. This was taken from another talk, and it has my 6 favorite hits. I won't keep you here. I'm only going to tell you about the first 3. One is going to have to do with a sequence that came out of Inder Verma's lab, VMOS. I'm going to tell you a long story about that, and the famous Rous sarcoma virus, VSARC. The second one, which is a short story in every way, but very interesting, has to do with a melanoma antigen coming from melanoma cells. And then the third one I'll mention is going to be about a platelet-derived growth factor. There were many of these. The astounding number of hits that were made is a function of what the biology was all about, that people weren't prepared for this. Of course, there were great disappointments. There were a large number of people who sent us sequences that they had laboriously cultivated out of something, and they turned out to be proteins that are in calf serum or various other artifacts that they had purified and were not really anticipating. One man thought he had cloned a particular polypeptide hormone. It turned out what he had gotten was ubiquitin. Well, the amazing thing was that other people had been trying to clone ubiquitin, couldn't. So this guy got it. This man literally changed his whole research program and became a ubiquitin expert. I mean, he just, it was fun. It literally was fun because the action was so fast. For an experimentalist, a lab experimentalist, it was heady stuff. Okay, well, I'm going to tell you about Inderverma's business, and especially he had a postdoc, Chuck Van Beveren. And this was all, retroviruses were becoming very big. This is 1980. And the standard description of a retrovirus was that out at one end over here, the 3' end, it had a transforming factor. And those were the guys that people were targeting. And DNA sequencing just getting going now. And Chuck was sequencing the transforming factor from this guy called BMOS. It was a mouse leukemia virus of some sort, a retrovirus. Well, they asked me to search it for him, and did, and ran it through our enormous database, which was such a drip at the time compared with now, but didn't find anything. I said, but we'll keep it around for you, okay? Fine. Well, I forgot all about it, and it was several months later that a journal hit my desk, and it had a sequence in it. I looked, and it was from a lab, famous lab in San Francisco, and they had a transforming virus. It was, in fact, SARC. And they had the sequence, I looked at it, and they had a footnote, and it said they had been given the sequence by Inderverma of the other guys, and they had searched it, and there was no resemblance. And I said, well, I'm surprised about that. I would have thought, these things, we'd hope was that they would have some common theme. But no matter what, I myself typed in the sequence out of the journal from this paper and put it in, and was set it on to search. Now, I must tell you how primitive searching was in those days. It's hard for the new generation to understand this. But we had a little computer, as I mentioned, it was a DEC PDP-11, and had very little memory. And so you had to take your output as you went. You couldn't keep things stored up and so on. So we had it attached to a typewriter called a DEC writer. And as it would go through and search, it finished one, it would type it out. I have to tell you, back up and tell you a story I didn't tell you. I had some marvelous undergraduates work on all this also. The best of whom was a young man named Neil Woodbury. And he set up all our early programs and so on. And he had the one who had set this DEC writer in motion. And we wanted to be able to look at an output and see immediately whether there was any kind of resemblance or not. It's like doing a blot. We were always going to get a lot of false positives. And how you set the stringency here. So there's always going to be a background. And we wanted to see the things that were really heavy. So he said, well, we could, thought about changing fonts. It couldn't be done, lowercase, uppercase. He said, why don't we leave spaces between all the characters, and every time that there's an identity, we'll put an underline, because the DEC writer couldn't back up. And so it would go, and when it sees one that's the same, the next one is an underline. So we call this sidelining. Now, I'm telling you all this because it put an audio into our life. As a thing, since this DEC writer was in my office there, when it was going along just doing routine printing out, it would go dot, 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 dot. But if it came to something that had a lot of identities, it would go dot, 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 because the frequency changed as it put those sidebars in. All right, well, I typed the sequence in, and a sequence search in those days for just the size of the database we had, which were now, be under 2,000, was an overnight search. Put it on. I was putting my coat on to get ready to go home, and I heard the DEC writer calling. Dot, 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 dot. And I went over and looked at the SARC sequence from San Francisco. It found something. And I looked. It had found Inderverma's sequence. And I looked at the paper, and the paper said there's no resemblance. But in fact, here is the output that came. Now, these were, they were clearly homologous proteins. How had San Francisco missed the boat? How were people thinking in those days? They compared the DNA sequences. They didn't change it into the proteins. They missed it. I mean, it just signaled a noise. I mean, it was, I'm sure I could hear the kicking of themselves later. But all the way down here. So all these things that turned out, they were so momentous, it was because we were all thinking differently. And it wasn't that there was some great innovation. It was just that learning how to approach these problems that were all around us. All right, well, later we obviously got into this, and these all turned out to be, of course, kinases. We did a whole bunch of these. A cyclic AMP-dependent kinase from a non-retrovirus was sequenced up at the University of Washington. The rest of these are all from various retroviruses, and it was obviously making a good story here about how these radiations occur. Okay, end of that one. Next one is quicker. I mentioned University of Washington. People were calling up now. A man named Joe Brown was at the Fred Hutchinson. And he said he heard we would search sequences against a database. And I said, yep, right. And I said, just put it in an envelope, send it down, we'll type it in. He said, couldn't I just read it on the telephone? And I said, how long is it? He says, well, it's 13 amino acids. I said, 13? That's pretty short. I don't know whether you really want to do this. You'll get something and you'll believe it and it won't be true. There's no good statistics here about 13. He said, oh, please. We did it. Anyway, he started reading the things. What they had done is they had been culturing melanoma cells forever, scraped off this antigen from the surface, got enough for one sequencer run with an Edmund degrading sequence, and had gone through 13 cycles. So I said, okay, I'm ready, read the first one. He said, well, the first one's blank. And it turned out that of the 13, they really only had 10 hits, but we searched it. And out of it came the fact that out of the first 13 residues, there were 6 identities, and it was with transferrin. Now the question is, was this true? Was this by chance or not? Well, Joe Brown was very good. Experimentalists, people that I really admire. They got themselves quickly some iron 59 and took their little bits of protein and found that, in fact, one of these is transferrin binding iron, the other is their melanoma protein binding iron. This little search of this little peptide saved years of work. There's no other possible way that they would have found the function of this protein. They didn't have any idea. It was just an antigen on the surface. Turned out, led to all sorts of experiments about how these cells feed themselves and take iron in and so on. Became known as P97 as the protein. It was very, very good. It just, once again, caught people's attention. Hey, there's something to this searching of sequences with computers that can be profitable to everybody doing it. All right, well, in 1983, wow, Science magazine carried an article about platelet-derived growth factor. Some 500,000 units of human platelets had been used in this study to get this growth factor out of platelets. They had 2 N-terminal sequences that came out of Edmund sequencers of the Edmund degradation type, and they were similar. Popped them in. It was easy to print these guys in. And this was Memorial Day weekend, 1983. I remember it so well. Here's the output that came. And in their paper, it said they had searched these, but I don't know how. But at any rate, against NUAT, it came out. There were 2 entries in there. They turned out to be the same thing. They were a sarcoma virus known as V-cis. So the platelet-derived growth factor had an overlap with this. Got it out. These were only partial sequences, remember. And got out the sarcoma thing and could see that this was an internal sequence that had been activated. There was a proteolytic site, obviously, there. And then the peptide fit right in there. It was amazing. Well, this was big news because everybody was interested in oncogenes. What were they? Were they really host proteins? Were they the seeds within you? Were they mutated? And so on. But here it was. Here was a genuine platelet growth factor and a genuine oncogene and V-cis, and they were the same. The differences, the capital letters in this case, are where they're not the same, is because this was from a woolly monkey and the other guys were from human, and so this was about the amount of change the primatologist would expect to be from between these 2 species. Well, what to do? Saturday morning, Memorial Day weekend, know all this stuff. There was no internet in those days. You could get on the telephone and call your mom, but I think that... So I thought about it, and I thought, well, I'll write to all the people involved. I'll write to the people who did the sarcoma virus, and I'll write to Hunker Piller and Antoniades, who did the PDGF, and tell them what they were, sent some photocopies over to the people at Salk who were my retrovirus consultants and things like that also. At any rate, wow, I could tell how fast the mail got delivered by how quickly the telephone rang. They compared notes, they exchanged antibodies, they were identical, big news, wow. Everybody was happy, oh, and so on. Then it turned out there was some guy going to throw a fly in the ointment. And the word got around quickly. One of the, we got a few silent partners decided they wanted to be on the publication. A big paper was going to be gotten together very quickly. And one guy giving a talk at a big meeting mentioned, oh, they found this. Sitting in the audience of that big talk was, in fact, somebody who was collaborating with somebody in England on the same subject. They were doing the sequence of platelet-derived growth factor. So this guy, who happened to be Tom Doolittle, who now works over here at the Scripps place, slips out of this talk, gets on the phone, and finds the people in England and said, the secret's out. What is the secret? They're doing the same thing. Well, big race. What happened here? Well, here I think, who is this? Waterfield. And it turns out that just 2 weeks before I'd gotten a letter from Mike Waterfield. And you can't read it, but I'll just tell you, he says, "Thank you very much for sending the tape containing your database and programs. It is a pleasure to receive such prompt, efficient, and free service. I'm sending a blank tape to replace the one you sent." Such generosity. I am. He said, "I hope I can be added to your mailing list." He said, "This service to the scientific community is so great." Well, he didn't tell me why I was getting all this praise, but of course, what they were doing, they had a sequence also. They searched it against our NUAT and found the thesis there. In fact, there's fastest publication I ever had. Science published this side's story in 4 weeks. Nature came in just 1 week behind with the Brits doing all this, and Tom Doolittle. But it was interesting, and once again, the value was that if you now bank it and look at it, it's worth more than just, it's not stamp collecting. These are things that are providing valuable information. So I'm just going to take just another 2 or 3 minutes now, next phase 3, to tell you what really sparked the field of bioinformatics. There's no doubt that the Human Genome Project really was a big spur to all this. And I thought you ought to know a little bit about the committee, but I'm also surprised that I realized that how naive I was to think that you could read all this. This is the membership of the famous Alberts Committee that was assigned to cogitate and worry about whether the human genome ought to be sequenced and whether Congress ought to give the money to get it going. And it was filled with heavy hitters. I mean, this was a committee of intellectual egos that you couldn't believe. I mean, if you read up there, I mean, Alberts is the chair, and he later became the head of the National Academy, and I think they gave it to him because they figured if he could handle this room full of intellectual bullies, I mean, really are bad. You have David Botstein and Sidney Brenner, you know, up there, and and down the bottom is Watson. Jim wasn't certain that we should do this. I mean, later he became the head of the project. There were people on here who were very much concerned that this was a waste of money. I'll tell you why they felt that way. There's one very important name that isn't on here, and that's Wally Gilbert, who was a member of the committee, but he got so disgusted with all the arguing back and forth that one day he didn't show up, and the next morning somebody brought in the Washington Post, and there's that big article, "Gilbert to sequence genome on own." And he literally started a company to do this. And I remember a quote from the reporter. The reporter said, "Can you patent the human genome, Dr. Gilbert?" He said, "No, but we can copyright it." So I don't know if that's true, that you can or not, but he said it, and it was in the newspaper. At any rate, I was on this committee, and Victor Mukusic and I were assigned to write a chapter for the report on evolution, which is what I thought would be the virtue of all this, that if you looked at a lot of things, then it would in fact be valuable, as opposed to just looking for getting a map of the genes. And the questions that were being asked, I won't read through all this, but were quite legitimate questions like, you know, will the introns matter? And will you be able, mainly the question was, can you identify the genes by looking at things like GenBank or not? There was one very eminent human geneticist who just believed it would be impossible to find the genes simply by gene searching against GenBank. He made a diagram that I have adapted here, but he just drew this on a little chalkboard, about the relative sizes of the human genome and the GenBank at the time. And he kept saying, how are you going to identify anything in here on the basis of that puny little thing over there? And others would say to him, well, of course, they say, well, only a small percent is coding, so we don't have to worry. But that's still bigger than the other, he said. But he missed the whole point. He was trying to think that sequences are infinitely random. And of course, everything I've been trying to tell you about this afternoon is that that's not true, that it's a great binomial expansion of gene duplications that are all related to each other, many close and many far, but they're all related to each other. They're not just combinations. He was stuck in one of those things like sequence space, where you think that there are 20 amino acids and there must be that many, if there are 350 amino acids, that there would be that many sequences. Or if you were, oh wow. Well, we missed in between here. In between it has domains with a number that's not quite as big as the large one. And the fact is, this is ridiculous, that none of that many sequences never has or ever will exist. It's much more confined and restricted space. The key is that where do new proteins come from? They come from the duplication of genes for old ones. Well, I have to make a few quick acknowledgments. I mentioned my kids, who really were the people I learned from at the beginning. I mentioned Neil Woodbury. Neil is a professor at Arizona State in biophysics, wonderful guy, terrific person every way. I mentioned Karen Anderson, who typed in scads of sequences over the years. And I haven't mentioned very much Dafei Feng, who's here today, and my graduate student Mark Johnson, both of whom in the 80s were terrific programmers as we went on to try and compete with all these other people who joined this field. And with that, I'm going to say this is the end.
Well, that was a terrific lecture, Russell. That was extremely interesting. To follow the history of any development like that from the beginnings to a state of considerable knowledge, and to be a part of it like that must be very exciting. Where do you think this whole area, proteomics and computerology and biology, is going to go next in the future?
Well, very hard to predict the future, of course, John. If we can look at the history, though, we can see that computers are going to get faster and faster and faster and be at everybody's tips. They already do much more than I think we need, considering how primitive everything was when we started. So there'll be more and more information, and it will be out there at everybody's fingertips. And I guess the concern is, how are people going to use the information? You know, in biology, now in certain areas of this, you don't have to do experiments anymore. The experiments have all been done. It's a matter of just mining the information that's there and collating it and put it together in novel ways in some areas. I don't mean that we're going to give up experiments.
There's going to have to be a lot of thought given to how to take thousands and thousands of bits of information gathered together and put them into some kind of systematic array where they mean something.
It is the challenge, but amazingly, people are rising to the challenge. I'm impressed with how many what we would used to call scientific contributions are now buried in the technology, and we take them for granted. It was 20 years ago, big deal to try to understand protein structure, for example, and how proteins folded up and so on. And now the computers have put so much, so many computer scientists have put so much hardware behind it all that anybody can go in and essentially do these things now. I don't mean to make it sound easy, but the fact is, what they have done on the other side of just providing machinery in the old days would have been a major breakthrough. And,
well, what new things do you think are going to come out of it?
okay, can I give you a couple of examples? All right. Now, the big deals right now are the major resources that are available on the net, the World Wide Web. So let's say that you're interested in trying to figure out where proteins came from, which is what I was really interested. Now, in the old days, you'd go into your laboratory, and when you were really primitive, you'd now try to get an amino acid sequence or a lot of things. Or later, you might try and get DNA sequences and so on. But now, you go in and sit in front of a computer terminal, and there are these banks and banks of everything there. Not raw information, it's already processed. So there's GenBank, has DNA sequences, and there's the PDB, has protein structures, and then somebody else has taken all those structures and parsed them into domains and families and superfamilies. And essentially what you have to do now is conceive of simple little routines for mixing and matching all these data. And people sit there and think about it, carve out a little niche, and the first thing you know, they have figured out where all beta proteins have come from, for example, and can make a family tree. All of this using software that's available right there on their computer. They had to have just, it's almost, in many ways, it just sounds like something that the Greeks would have thought, well, once we had this, we'd understand everything in the universe. Even with all this power, there are fundamental mysteries, and I worry whether they're going to fall easily. The downfalls of all this system is that people are becoming increasingly specialized. The people who do the computer science are going more and more into their little niche. The people who do the technology of sequence gathering are more and more into theirs. We're not educating our students awfully, what you might call broadly and capably. In other words, it's one thing to be broad, but usually the kind of thing we say broad is very superficial. But if you want to be broad and capable and get to the roots of things, not a lot of that happening.
This information may be accessible and meaningful to those people who can do that kind of thing. Meanwhile, the average man in the street in the year 2060 who's still rebelling against having his son taught evolution will not be the person who will be able to take advantage of that new information. It's as if the information is becoming rarefied at the same time that it's becoming so complicated and all-knowing. So.
Yeah, but now we're into another realm,
Yes.
And that is that here we have all this knowledge, and we're not dispersing it well at all. Television has been a great disappointment. I mean, even the so-called public television, now just riddled with commercials at every step, doesn't nearly measure up to what it could do with science at any level.
What the public wants. It's what the public wants.
Well,
so that's not so. It's not so simple as that.
No,
we're not doing the right thing.
We know all this, but we're not teaching it effectively. We're not giving anybody a chance to learn it. Now, when we think about what public school system in the United States used to be really a wonderful thing. I mean, you and I are both public school graduate types that have gone through all this. And today, the public schools are in bad shape because the public refuses to pay for them or even acknowledge that they need paying, and it's just ruinous. The colleges aren't doing much better. It's been known for the last well, ever since you and others got going on setting up UCSD, that the sciences in most places are really secondary, and they're not nearly enough taught, and certainly not taught well, and it hasn't improved here. And so I feel that we have all this knowledge, but the dispersal of it is just terrible. It's just not really. We've sucked a lot of people into biology as graduate students. They can't get jobs in the academic world because there are so many of them, but they'd be wonderful secondary school teachers if the system would provide for them properly and not turn them into overworked disciplinarians and some sort. Many of them wouldn't mind teaching secondary school at all under the right circumstances. So the social problems have just overwhelmed the scientific here and ruined it in many ways, I guess. Hey, this is a pretty dismal conversation we're having here.
I remember that you were a high school teacher at one time.
I did start off that way. And the interesting thing is that the reason I went to graduate school is that I felt that I was so phony as a teacher that I simply didn't know enough biology. Trying to keep ahead of them in a very bad textbook. And now, of course, many years later, I do feel as though I'm equipped to teach high school biology finally, and I don't really have the energy. But I wouldn't mind it at all. I think I really have a grasp of it now and could do the job well enough.
Well, good luck, Russ. I hope that you get your opportunity. We've been talking with Russ Doolittle, Department of Chemistry and Biology at UCSD, and I've got the final word.
Okay. Well, that